Okay, we're going to go ahead and get started here. Thank you very much, everybody, for joining the first session of Virtual Podium. Our session today will be focused on general metabolomics and lipidomics applications. A little bit about ourselves. So Virtual Podium was actually established to promote scientific discussion and engagement during this very challenging time in our global history. And so recently, many of us have been impacted by the cancellation of conferences. And so not only are we losing the ability to present our research and share that research, but also the opportunity to connect with our community. So a group of us decided to pull together and present here to you the Virtual Podium Symposium, which is actually a platform that allows you the opportunity to share and present your scientific research. And we targeted this to be focused for mass spectrometry-based biology. And you can see here in the illustration in the bottom right-hand corner, the types of topics that we hope to target. And so using the virtual platform, we're really creating a sense of community and providing the opportunities for you to be able to present your work and learn more about the compelling scientific research that is happening in these fields, particularly centered on chemistry, biology, and informatics. And many of us had some of this content all ready to go. And like I said, because of those canceled events, we've decided to create this actual podium to allow that engagement. The topics are going to be changed weekly. We're going to meet every Friday, and we'll hear for 5 minutes from each of our lightning talks from researchers across the globe. And those topics will be suggested by you, the scientific community, and so you have the ability to do that at the end of today's session. I'd like to present the team that pulled this together. My name is Baljit Yubi. I'll be your host today and present the speakers. I'm a senior market development manager for life science research business at SciEx. My co-hosts here today are Aparna Vania. She's a postdoctoral researcher from the UC Davis Metabolomics Center. Ivana Blaznovich, who is a senior metabolomics scientist at DiscernDx. Christina Jones, who joins us as a research chemist from the National Institute of Standards and Technology, and Alice Darlow, who's a staff research associate at the UC Davis West Coast Metabolomics Center. This is a brief overview of our agenda today, and just a note for you all, we have had one speaker drop out, unfortunately. Katie Love's talk is now going to be swapped out with Aparna Vania, who has kindly stepped up at the last minute to deliver her talk. So I'd like to welcome first and foremost our keynote speaker, Professor Oliver Fiehn. Professor Oliver Fiehn is from the West Coast Metabolomics Center and has pioneered very many developments and applications in the field of metabolomics with over 220 publications to date. Since 2004, he's been overseeing the research laboratory at the UC Davis Genome Center, which also serves as a satellite core service lab in the metabolomics research space. And since 2012, he served as the director of the West Coast Metabolomics Center, supervising over 30 staff and over 16 mass spectrometers, coordinating with 3 different satellite sites. Professor Fiehn's research aims at understanding metabolism on a comprehensive level and works in many different scientific domains. He actually collaborates with a range of investigators in studying human diseases through statistics, text mining, and pathway mapping efforts. For his work, Professor Fiehn has actually received a large number of awards, including the 2014 Molecular and Cellular Proteomics Lecture Award, and the 2014 Metabolomics Society Award, Achievement Award. He serves on the board of directors for the Metabolomics Society from 2005 to 2010 and 2015 to 2017. amongst other capabilities, and serving on many organizing bodies for certain conferences. And also, I've got to give Oliver a shout out for really being our champion for the virtual podium and helping us be endorsed by the Metabolomics Association of North America. Oliver, I'll hand it over to you now.
Thank you so much. It's really my pleasure to see that so many people have gathered in these times. In fact, times have been weird over the last years, as we have seen, because I think science is under attack from many sources. More and more people, say that facts don't matter, and only opinions matters, and feelings matters, and I believe as a scientist we have a duty to stand up for the truth and look at the facts and see where the data lead us. And here you can see our laboratory in the March for Science in April 2020 of 2017 in front of the Capitol in Sacramento. So our laboratory is very much on the forefront of data science and producing good data. Next slide, please. Okay, so in our laboratory, we have done metabolomics over 20 years now, and steadily improve our abilities to identify compounds, to annotate compounds. On the left-hand side, you see Dr. June Ding's research. She was here, a postdoc, until January, when she left to Wuhan, the center of the epidemic in China, because she wanted to be with her family. And we see 2,000 identified compounds in different brain regions. We'll publish soon about the impact in biology. On the right-hand side, you see Arpana Bania's research, also identifying and annotating over 2,000 compounds in different platforms in the US National Produce Initiative, from tomato to garlic and strawberry and basil. And as you can see here, for example, we identify over 918 different natural products on a single column. And of course, when I get these data and I see these reports, and similar reports have been published by companies, I always wonder, how certain are we when we present these types of data, and how can we make sure that other people understand what we are talking about and can verify it? Next slide, please. So, people used different levels of certainty, and the Metabolomics Standards Initiative had, suggested some levels, and new levels have been suggested. But when we look back at it, we need to think about that any compound annotation relies on multiple lines of evidence. So, for example, in the realm of chemistry, we not only have an accurate mass and isotopes and adduct abundances, but certainly we have mass spectral matching to various types of databases and libraries. But that is not all. We also have retention time and collision cross-section through ion mobility. All these chemical avenues lead us to more and more confidence. And all these are data that need to be reported if people annotate compounds. In addition, we now realize we cannot have 100 million compounds. We need to have a lot more in silico predictions, again, for both MS spectra, retention time, CCS, and also NMR. But even that is not enough, because we understand that there have been people before us. So many papers have been published. So if I know what I'm looking at, if I'm looking at garlic, it makes sense to see what has been published already in garlic and in other resources. Next slide, please. For that reason, that doesn't that doesn't show anything here. No, okay, well, that doesn't present well. Well, what you cannot see here are these actual slides that show a little bit on the data. It's okay, Bal. Don't worry about it. Don't worry about it. Bal, now now it's gone. Okay, well, it doesn't matter so much. What I wanted to say is that my laboratory has weighed in over time and produced a number of different tools. Maybe the most important tool is massbank.us. We also call it Massbank of North America. It's freely available, it's public. It stores over 600,000 spectra from all over the world. So we have collaborated specifically with Riken Hiroshi Tsugawa and Masanori Arita and others in Japan, but also with David Wishart's lab in Canada, also with GNPS, Peter Dorstein's lab in UC San Diego, and many others. So the idea is that all experimental spectra that are freely available can be looked at and downloaded at massbank.us and can be sorted by metadata, for example, retention times if this is uploaded, certainly the different types of mass spectrometers that are being used, and so on. We have also, with Hiroshi Tsugawa, published a number of new software, for example, MS Finder, that enables to predict spectra, and we are constantly working to improve that. We are now also going for different types of chemical analyses where we get more information of unknowns, for example, the number of acidic protons, where we use proton-deuterium exchange and LCMS. But we also look into as I said, the realm of biology. We published the bloodexposome.org database with Dinesh Barupal that basically looked at over 28 million abstracts and 6 million full papers. And we found almost 1 million papers that reported chemicals detected in blood. And overall, these were 42,000 chemicals. So that gives a good a priori information of what is expected when we look into blood. Similarly, in my laboratory, we have investigated GCMS spectra over 20 years in over 2,500 studies that you can look into, investigate which compounds have been found in which organ, especially also for unknowns. You can upload your spectra and see if the same spectra and the abundance of those compounds in different organs and matrices. So the idea is now that all these different lines of evidence can be combined in terms of error models, false discovery rates, weights, how much weight do we give to an accurate mass, and how much weight do we give to matching MS-MS spectra, to come up with ultimate scores of confidence that our reported metabolites are correct. Next slide, please. Okay, here we actually see things. We think that it's great to have good data, but you need to be able to see those data. And we regularly upload our studies and reports in the Metabolomics workbench. That is an NIH database, and on the top you see that we do this all the time. There's accession numbers, and this is a study, for example, in mouse, where we looked at a number of mice knockouts, and anyone can download those. And indeed, Paulo Bonini in Tarragona, Spain, made use of all those public data. He downloaded the retention time libraries from Mass Bank of North America. Later on, we downloaded also the data from the mice, from the knockout mice. And what Paulo did is he developed machine learning methods, and he had 5 different types of machine learning methods for both reversed phase with Hiroshi Tsugawa and also Hillig with us. And we can use this kind of retention time modeling now to remove false positives. And on the right-hand side, you see a couple of false positives where the MS-MS spectra were very good. We're really matching nicely with a number of compounds. And you would look at the MS-MS spectra and you'd think that they would completely be identified. But if you then look at the observed retention time and the prediction retention time, you see that they often have 2 minutes or even 5 minutes of error. So we can remove them through this additional type of metadata. Next slide, please. And I would like to encourage everyone here on the call to upload your data to FAIR databases. FAIR databases are findable, accessible, interoperable, and reusable using persistent identifiers, rich metadata, public use, usage licenses, but also ability to look at the provenance of data streams, using FAIR vocabularies and ontologies. And we certainly need to improve that as a community. And in order to make data available to the public, let us find the truth in data. And with that, next slide, please. I would like to thank the West Coast Metabolomics Center team, specifically Jun Ding in China, Paulo Bonini in Spain, Hiroshi Tsugawa in Japan. And in this slide, you also see Arpana and Ivana Blazinovic, actually, and a couple of others who have contributed to this research. Thank you so much.
Okay, thank you very much, Oliver. I really appreciate that talk. And in the interest of time, we're actually going to save the questions for the end of our speaker presentations. So if you have any questions for Oliver and the following speakers who are going to present their posters, please do use the Q&A box to submit your questions, and we will open up the lines for 30 minutes of Q&A so you can ask Oliver and any one of the speakers here any questions about their work. Thank you, Oliver. Okay, so with that, I'd like to introduce our first speaker, who is Natasha Machado. She's here from the School of Medicine at the University of Sao Paulo. Over to you, Natasha.
Hello, everyone. Thank you very much. I'm going to share some part of the data I had from my PhD. The title is Type 2 Diabetes Metabolic Improvement After Roux-en-Y Gastric Bypass may include compensatory mechanisms that balances fatty acid beta and omega oxidation. So what I'm showing here today is just a part of the research, and I'm going straight to the point. So we know that the origin of insulin resistance in obesity and type 2 diabetes is not limited to impaired insulin signaling, but also includes complex metabolic interactions, imbalance of metabolic regulation, and alterations of cellular functions. And components of insulin signaling pathway, such as lipid oxidation, lipogenesis, hepatic gluconeogenesis, and protein synthesis and degradation, they may be regulated by metabolites, especially through alterations of substrate flux. And Roux-en-Y gastric bypass is a type of bariatric surgery and is associated with early improvements in glucose homeostasis. But the mechanisms underlying this condition are still not fully understood and still requires consideration. So by analyzing gastrointestinal and plasma samples through metabolomics, our goal is to characterize gastrointestinal and systemic metabolomic response to Roux-en-Y gastric bypass and their contribution to metabolic adaptations associated with glucose homeostasis. We know that Roux-en-Y gastric bypass was associated with systemic and gastrointestinal metabolic reprogramming, as you can see in the principal component analysis. And by looking at the metabolites, this was significant in all sample types except in the stomach median body, which is highlighted in red. So metabolite alterations, they were associated with important changes at the chemical and pathway level, and particularly decarboxylic acids, they were relevant in several metabolomic tests. So the product-substrate metabolic relationship between these molecules suggests that decarboxylic acids alterations, they link omega and beta fatty acid oxidation to homeostatic mechanisms, including the tricarboxylic acid cycle improvement. So we hypothesized that these might serve as a metabolic compensation to mediate gastrointestinal and systemic changes in glucose metabolism, because we know that mitochondria beta oxidation is impaired in type 2 diabetes, and Roux-en-Y gastric bypass may increase metabolic flexibility by redistributing fatty acid oxidation between endoplasmic reticulum, where the omega oxidation occurs, and peroxisomes and mitochondria, where beta oxidation occurs. So the metabolites produced by omega oxidation, these decarboxylic acids, they might be further metabolized by beta oxidation, producing increased levels of succinic acid, which is also a decarboxylic acid, and this is an epimetabolite that can help to improve tricarboxylic acid cycle as an intermediary metabolite. So I guess this is it. I hope it's clear for everyone. Thank you.
Okay, thank you very much, Natasha. And we really apologize for losing some text in the top right-hand corner of your slide there, which we'll substitute in the recording at a later stage. Thank you, Natasha. We'll move to our next speaker. So our next speaker's name is Saiful Khan. He's here from Elucidata in India. Over to you, Saiful.
Thank you, Val. Hey guys, I'm Saiful. I work at Elucidata on developing L-Maven. L-Maven is our metabolomics data processing tool. And we recently landed some SWOT capabilities in L-Maven, and that's what I'm going to talk about right now. Now, data independent acquisition, that is DIA, is where you fragment your precursor ions within an M by Z range and then record full scan spectral information for all your fragments. We do this for the entire M by Z range of interest to get a thorough spectral profile of our biological sample. SWOT acquisition, which is a type of DIA technology, has found a lot of use cases in proteomics, specifically quantitative discovery proteomics, and with some recent improvements in tooling in metabolomics, such as MS-DIAL and MET-DIA, it's found some adoption in metabolomics as well. But a broader adoption remains a challenge because the existing tools are prohibitively time-consuming, and that's where L-Maven comes in. Now, L-Maven is a vendor-neutral and cross-platform software. It was originally developed at Rabinowitz Lab as Maven, but since then, for the last 3 years, Elucidata has optimized and enhanced Maven as L-Maven, and we are also committed to building a community around this open source project. At this point, you can load gigabytes of sample data and run large compound databases and do automated peak detection and reach your result table within minutes. Our new SWOT method has a deconvolution algorithm that generates a purified spectra for your precursor ions, which you can match against any user-specified spectral library. And we accept MSP and MGF formats because they're both open standards for representing spectral information. And more importantly, this method is really fast. So we did some preliminary analysis and compared it. So for an experiment with 24 samples, L-Maven, we found that L-Maven is 25 times faster than the most popular open source alternative, which is MS-DIAL, I should mention. And I should also mention that MS-DIAL was really helpful in developing our own method because we took a lot of ideas from their work, and specifically the deconvolution algorithm is working really well. So thanks for that. But we're continuing to improve on that, and we're not just aiming for speed. So L-Maven is also integrated with Poly, which is Elucidata's cloud platform. So once you have your annotated metabolites, you want to push them to Poly and perform some downstream analyses, such as differential expression, metabolic flux analysis, and a lot more things. To add to that, we provide technical support to our users, as well as on-demand training, should you feel the need to dive deeper into how it works and understand all the algorithms, things like that. Last but not the least, Elme is open source. We have a GitHub, we're on GitHub. So our feature board is always open. We welcome any sort of discussions, issues, requests, especially because our new method, this SWOT method or DI method is in its infancy. We'd like to hear from the broader community what it is that they expect from a DI workflow, so that it helps us shape the tool to serve them best, right? So please reach out. Thank you.
Thank you, Saiful, for keeping to time. I really appreciate it. And we'll move to our next speaker. Our next speaker is Tong Sheng. She is from the West Coast Metabolomics Center at UC California in Davis. Over to you, Shen.
Hi, thank you. Thank you for the organizers that put together this virtual podium. And I'm going to use this opportunity to talk about our team's effort to make use of the microRLC coupled with tandem MS for untargeted metabolomics analysis. In this case, developed a fast and very sensitive method to analyze natural products in common food products. The natural products, including polyphenols and flavonoids, because of their very good antioxidant potential, are actually quite important compounds in food research, plant metabolism, and traditional Chinese medicine, so on and so on. And it has a quite vast complexity. And here what we wanted to do is evaluate the benefit of using the microRLC column that is usually 0.3 millimeter inner diameter to 1 millimeter instead of the classic 2.1 and higher millimeter UPLC columns to analyze like a wide array of metabolites. In this case, it's the natural product. And the microRLC column here have been shown before to greatly improve the sample throughput and sensitivity. And here what we make use of is the 0.5 millimeter kinetics F5 column, and also the SciX M5 microRLC microfluid LC system. And together, hand in hand with the OptiFlow ion source coupled with the TripleTOF, because the 1 point of microRLC really have to reduce the void volume like after like coming out of the column and right into the ion source. So we do have like a UPLC method developed in the lab before that is based on a PFP column, pentafluorophenol column. And it's a 10 minutes analysis UPLC, which with the flow rate of 400 microliter per minute. And we adopt the same gradient, same mobile phase, but swap that to microRLC scale. So the flow rate is, instead of 400 microliter per minute, we're using 50 microliter per minute. And the column is very similar to PFP column, which is F5, but a microRLC column. In the end, we compare to the classic 6 to 12 second peak width for UPLC. We obtain like average 2 to 3 seconds chromatographically separation peak width using the microRLC. And the TripleTOF luckily is very fast, speedy to catch up with our MS1 and data dependent acquisition on MS2. So first, of course, we wanted to test like using mixtures of synthetic standards for a wide range of natural products to test the separation and sensitivity. In terms of that, we actually tested 26 groups of natural products with like more simple modifications to the 3-ring structure to the more complicated modification. Most of the compound, most of the group, we obtain similar separation power, like resolving power between UPLC versus microRLC. Their number of theoretical plates actually are similar, but we do have like a few pairs that in microRLC give us a better separation, which is actually in my lower left figure. The upper one is the UPLC result separation, and the lower one is the microRLC one. It is based on the pentamethoxyflavones. And in terms of sensitivity for this pair of isomers, we also get like 50, 60 more higher sensitivity, higher signal compared to the UPLC. And using the epigenein deuterium label, that's one of our internal standard, to test on the sensitivity as well. We also see like a 22 higher signal to noise ratio increase used in our micro-LC. Stepping forward, then we use this untargeted analysis to analyze food metabolites using this wine, green tea, and coffee extract and yeast bilberry and sample for that. In the end, we get higher M over Z feature, higher number of M over Z features, comparable number of acquired MS2 spectra, and more or less the same number of annotations between the classic UPLC and micro-LC. And that could be, actually our cycle time can be further improved to get high MSMS. And also there can be the limitation of how many reference MSMS we have out there to do the identification, and which is also another project ongoing. So with this top 10 DDA and research based on MONA and NIST-17, we get a overall, this is first round DDA, we didn't do iterative back then. And we did overall like 260 identifications across different matrices. And the metabolites, if you see this pie chart here, metabolites radiated from flavonoids and polyketides, lipid molecules, organic acids, and biogenic amines, or sort of that. Most of like 150 we get from positive mode, and with extra 100 annotations from negative mode. And most annotated metabolites are very specific to each of the food analyzed, which is expected, and they are not much shared between two or more foods. So overall, we showed there is an excellent separation with a higher peak capacity with our micro-LC method, but with much shorter runtime, like half of the runtime, overall is 5 minutes, and narrower peak width, with improved overall sensitivity, at least 10-fold. Some compound we see 100-fold sensitivity increase from classic UPLC to micro-LC. And this method could greatly aid our profiling of natural products in food and other similar materials. Thanks.
Thank you, Shen. Thank you so much. We'll move to our next speaker here. Our next speaker is Nicola Zabalegui. Hopefully I did that surname some justice. And Nicola is presenting here from the CIBION Institute. Over to you, Nicola.
Hello everyone. I'm doing my PhD at the Bioanalytical Mass Spectrometry Group of the Center for Bio-Nanoscience Research, with the supervision of Dr. Maria Eugenia Monge in Buenos Aires, Argentina. The study I'm going to present was developed within the frame of the Metabolomics Quality Assurance and Quality Control Program led by the National Institute of Standards and Technology of the US. This interlaboratory study focuses on supporting measurements comparability of untargeted metabolic profiling in human plasma, and aims to find out a consensus characterization of metabolites present in candidate reference materials using multivariate statistical analysis. The suite of candidate reference materials comprises 4 pooled plasma samples from human donors, including diabetic plasma, high triglyceride plasma, young African American plasma, and normal human plasma. To accomplish these goals, we have used a reverse phase UPLC QTOF-MS based method to analyze the samples following our QA and QC practices. You can visualize a scheme that summarizes the sample prep protocol. I will only highlight some details. At the beginning of the experiment, each plasma sample was split into 6 aliquots. As well, a pooled QC sample was created from equal aliquots of each plasma sample. The following procedure was performed for each plasma aliquot to obtain 6 replicates preparation, 6 preparation replicates for per sample class. A mixture of methanol and acetonator was used for protein precipitation. In the middle of the slide, there's a simplified scheme of the sample list used for analysis. The initial injections were used to reveal problems such as possible impurities or contaminant presence in the LC system and during the sample preparation steps. Next, system suitability sample comprised of a mixture of 8 chemical standards was used to assess M/C, retention time, peak area, and mass resolution reproducibility over time. After that, 8 pool QC injections addressed as system conditioning QCs were used to equilibrate the analytical platform. 3 pool QCs consecutive injections performed after the system conditioning and at the end of the batch were used for locally estimated scatter smoothing, batch correction, lowest, a tool used to account for the time-related systematic variation of metabolite responses. After these QC injections, samples were randomly analyzed within a template of pool QC samples. This block of 4 samples bracketed by pool QCs was repeated 5 more times, being each block individually balanced and randomized. Data preprocessing scheme shows different curation steps that were performed after data import, chromatogram alignment, and the subsequent peak picking process, which was done with Progenesis QE software. The data preprocessing was carried out using an in-house pipeline implemented in the Python programming language. The first step was implemented to remove features with retention time lower than 60 seconds, as the system dead time is about 0.8 minutes. Second step allowed removing features that were not present in all QC samples as a condition for performing the lowest batch correction. After that, we perform the blank correction and we apply a filter based on the coefficient of variation. That is, all features with a CV larger than 20% in pool QC injections were eliminated. Subsequently, we applied a 100% prevalence filter and finally the D-ratio filter, which was used to remove features with 0 or low biological information, leading to the final data matrix. Regarding data analysis, after removing low quality data from the dataset, principal component analysis was performed on autoscale data. As you may see, pool QC data points cluster tightly in the middle of the PCA scores plot, which is a rapid check of data quality after the curation process. A genetic algorithm feature selection method was used to obtain a reduced panel of 20 features capable of sample classification. The best PLSTA model with the lowest root mean square error of cross-validation was selected, and a new PCA model was built after that. This new PCA model shows 4 sample clusters in the scores plot. This reduced panel would allow quickly quality assessment in the future studies and also across different laboratories. Currently, we are working to identify these 20 selected features. Thank you for your attention and for this opportunity.
Thank you, Nicola. Any questions for Nicola and our previous speakers, please use the Q&A box to submit your questions. Okay, our next speaker is Aparna Vania. She is also from the NIH West Coast Metabolomics Center. She is actually subbing in for Katie Love's talk today. Unfortunately, Katie was unable to present, so Aparna will be presenting. Over to you, Aparna.
Hello, thank you, Bal. My name is Aparna Vania, and I'd like to talk to you about the study that was done at UC Davis, West Coast Metabolomics Center. It's a comprehensive metabolomics study in 2 mouse model types for hypertrophic cardiomyopathy. So if you can see here on the left-hand side in the introduction box, what HCM is dealing with is it is a genetic disorder, cardiac disorder, that affects the thickening of the heart wall. So essentially, we're having trouble with pumping blood because the muscle in the heart has increased in terms of how thick it is. So these are the 2 types of mouse models that we are currently using. Each have a mutation in the type of muscle that is in the heart. So what we've done here is that we've taken 64 mice hearts, both from mutant mice and control mice, and used a comprehensive approach in trying to analyze and understand this genetic disorder. So we have both untargeted and targeted metabolomics platforms. Here you can see in this upper right corner here, we have complex lipids, primary metabolism using GC-MS, biogenic amines, and also targeted platform for NADPH. So with this, what we have found is that we are able to analyze and detect over 900 metabolites in these mice. And from the lipidomics platform, what we've started to learn and started to discover is that in the TNT mutant mice, what we're seeing here in this Kemrich plot, we can see that the carnitine levels are significantly decreased, which is a very good indicator that the mitochondrial function is impaired. So using charts like this allows us to really assess our understanding of this genetic disorder, and that's really all I have for you. So I'd happily take questions at the end.
Okay, thank you so much, Arpana. I'm going to turn it over to our next speaker, who is Crystal Concepcion, and she's here from the University of Illinois, and I'm going to hand it over to you, Crystal.
Thank you, Bal. Okay, I have a question. Does anyone here like weeds? Okay, well, I'm sorry to say I do not like weeds, and I'm here to tell you why. Okay, weeds such as water hemp or Amaranthus tuberculatus are some of the most problematic weeds affecting agricultural production. Water hemp is an annual dicot weed that has evolved resistance to several commercial herbicides, inhibiting the 4-hydroxyphenylpyruvate dioxygenase or HPPD enzyme. Previous studies using commercial herbicides such as triketones or pyrazoles have demonstrated that rapid oxidative metabolism of the parent compound confers resistance in multiple herbicide-resistant water hemp populations. So as we believe that this resistance is really troublesome, of course, and as we are trying to minimize crop loss. So the goal of our research is to investigate the resistance mechanism for a non-selective HPPD inhibiting herbicide called syncarpic acid 3, or SA3, in 2 resistant water hemp populations compared with 2 HPPD inhibitor sensitive populations. SA3 is presumably metabolically blocked, or it cannot be degraded. However, early dose response experiments show that SA3 might be metabolized by certain type of weeds and not by others. Interestingly, certain resistant water hemp populations, such as SIR, show less injury to this compound SA3 compared to existing HPPD inhibitors in the market, despite not having been exposed to this compound. So theoretically, this means that SIR has not even evolved resistance to SA3 yet. To determine whether this is true, we treated excise leaves of resistant and sensitive water hemp populations with SA3 and quantified the amount of the remaining herbicide at 2, 4, 8, 12, and 16 hours after treatment. As it turns out, water hemp populations not only look different, but also metabolize SA3 at different rates. We found that the resistant SIR and NEB populations exhibited faster rates of SA3 metabolism, particularly at 12 and 16 hours after treatment compared to both the sensitive populations, ACR and SEN. But the next question that we ask is, what has happened to the metabolized SA3? Where did it go? To answer, we carried out untargeted metabolomics of the excise leaves from SIR, which is the resistant population, and ACR water hemp populations. To determine whether SIR is different from ACR, we performed multivariate statistics, which showed clear separation between the 2 populations and between the time points of harvest. Again, we verified that SIR can indeed metabolize SA3 much faster than ACR, and that the overall metabolite profile of these 2 weeds are distinct. Although some of the metabolites were reputatively identified, the majority of the compounds were unknowns, unfortunately. Among the most interesting and discriminatory metabolites was a putative hydroxy SA3 that was more abundant in the resistant SIR population. For our next task, we look forward to characterizing the metabolites formed in SA3-treated leaves at longer time points and using whole plants. And also, we like to deduce possible metabolic enzymes involved in SH3 detoxification reactions. So now, remember the start of this talk, I asked if anyone here likes weeds. Maybe now you know why I don't. Thank you very much for your attention.
Thank you, Crystal.
Is it 4 minutes?
Okay, next talk here. Our next speaker is Annalise Poss. She's from the University of Utah University of Utah Nutrition and Integrative Physiology. Over to you, Annalise. Annalise, we can't hear you, so if you could just unmute yourself.
Sorry about that.
No problem, we can hear you now. Go ahead.
Great. As I said, I'm a second-year PhD student at the University of Utah. My name is Annalise Poss, and I'm in the lab of Dr. Scott Summers in the Nutrition and Integrative Physiology Department. In the Summers lab, we study a subset of sphingolipids, termed ceramides, and their role in driving cardiometabolic disease. A large body of literature and preclinical models implicate specific ceramide species in driving cardiometabolic disease. And in the last decade, a number of papers have been published profiling serum ceramides in human patient populations and associating increases in serum ceramides with insulin resistance and cardiovascular disease, specifically coronary artery disease. In the study I'm presenting to you today, we aim to capture a more diverse sphingolipid panel than ceramides alone and determine if a more inclusive sphingolipid score would display an even tighter association with disease. To get at this question, I have this schematic that summarizes our study. We performed liquid chromatography tandem mass spectrometry-based targeted lipidomics on samples from the Utah Coronary Artery Disease Study, or CAD study. This consists of a healthy population controls and CAD cases from Salt Lake City, Utah, and we ended up extracting samples from 674 available biospecimens and collecting data on 32 sphingolipid species. Next, we applied a machine learning approach as a variable reduction, both random forest and lasso approaches, to generate a predictive algorithm termed the Sphingolipid Inclusive CAD Risk Score, or the SIC score for short. We also calculated a previously published ceramide-based score, the CERT1 score. We found that 30 of 32 sphingolipids measured were significantly increased in CAD patients, and this can be seen from this representative box plot for C16 ceramides here, with the levels being considerably higher in cases than in controls. We also found, which is not shown here, that the 30 significantly upregulated lipids displayed robust odds ratios for CAD as well. This next figure, you can see a Gaussian graphical model for the analytes that were measured. The lines represent conditional correlations above a threshold of 0.2, with the thickness of the line denoting a relationship, and the thicker the line meaning a stronger correlation. The color denotes the direction of the relationship, with pink as negative and blue as positive. Not surprisingly, as they're in the same biosynthetic pathway, the sphingolipids conditionally correlate with one another, forming this ring. And other conventional lipid markers, such as cholesterol fractions and triglyceride, do not, and they are seen separate here. This is critical in biomarker development because this tells us that sphingolipids are conditionally independent from cholesterol, suggesting that these are not redundant biomarkers, but rather are tools that allow us to assess risk previously uncaptured by our clinical lipid markers. And lastly, I will show you this ROC AUC curve comparing the SIC score in black here, this top line, to other conventional lipid markers. So as you can see, the SIC score is shown by a black line, and it outperforms LDL cholesterol, triglycerides, and CERT1, which are represented by the other lines. Red here is LDL cholesterol. This line is a little hard to see, but this is the CERT1 score. And then triglycerides are nearly aligned with the line of no difference. This shows that a purely ceramide-based score may not be the most sensitive biomarker, but rather capturing rates of flux through the de novo synthesis pathway through the use of a more inclusive sphingolipid score may provide a more sensitive readout of risk. Moving forward, we plan to measure our sphingolipid panel in more diverse human cohorts to better understand the relationship between sphingolipid levels and human disease. So I'd like to end this by thanking the virtual podium team for the opportunity to be here and for all of you for taking the time to listen to my talk today. Thank you.
Thanks very much, Annalise. Keep the questions coming, everybody. You can use the Q&A box to submit your questions. Our next speaker is Marinka Tarantino, and she is from the Foundation of Edmund Mack at the Department of Food Quality and Nutrition, and the San Rafael Scientific Institute Center for Omics Science in Milan in Italy. Over to you, Marinka.
Thank you very much for the invitation, for acceptance of this talk. So I will tell you a little bit about the apples and the health. Some time ago, University of Reading executed a nutritional clinical trial with 40 mildly hypercholesterolemic volunteers who consumed 2 apples per day for 8 weeks, or as a control, they were drinking a sugar and energy matched beverage. The population was a free living population, while the apples were added to their habitual diet. You can see the experimental design on the left side of the picture. And as you can see, several types of biological fluids were taken during the study. Blood, 24-hour urine pool, and the feces. This allowed us to perform a broad and a comprehensive analysis, including different omics assays. And among them, untargeted LC-MS analysis for urine and plasma were performed. Also, the quantitative analysis of bile acids in plasma, and finally, metagenomics in feces. Of course, except of this, all the typical clinical measurements were also performed, such as cholesterol, for example. And I will stick to the metabolomics, untargeted metabolomics assay, which I think is the most exciting one. So the untargeted LC-MS assay revealed several very interesting and exciting endogenous and exogenous metabolites being statistically significant after the apple consumption, meaning that I want to say that they were upregulated. For example, hydroxyphenyl acetic acids metabolite were found, and they belong to the tyrosine pathway. And you can see the picture on your right side with the pink square showing you the tyrosine and the other metabolites belonging to the pathway that we found in the urine. Other very interesting family included indoles and the tryptophan that, of course, belong to the metabolic pathway of tryptophan. What we did next was to take all the metabolites being statistically significant, and we correlated them with the outcomes of metagenomic analysis. And there are several interesting correlations were found. For example, the one between granulicatella and tyrosine metabolites. And you can see the zoom and the screenshot of the heat map showing you those results. So in general, the identification of polyphenol microbial metabolites suggests that the apple consumption mediates significantly the good microbial metabolic activities, and this should be definitely further explored. Except of verification what happened in the human body and to metabolome of plasma and urine after the apple intakes, we were also approaching the second topic, which was the validation of fluoretine metabolites as the BFI. BFI stands for the biomarker of food intake. And we confirm the role of fluoretine metabolites, fluoretine gluconoride and fluoretine gluconoride sulfate, as a promising biomarker of apple intake. We evaluate several criteria such as the plausibility, the specificity, the time response, and the dose response, and we successfully accomplished the evaluation of such a criteria. So if you would like to know a little bit more about this study, you can click on the link below the 2 machines that are on the pictures. There is a DOI link. If you have any questions, you can ask later on, or drop a line, or contact the owners of the study, which are mentioned also in the slide. Thank you very much.
Thank you, Marinka. And now to our final speaker, who is John Laycock. He's here from the company TCAN. And so, John, I'll turn it over to you.
Hi, thank you. Thanks for the opportunity. And I'm going to talk about sample preparation. This is a fundamental problem, I think, at least in proteomics, and I would mention my talk today is covering a use case in proteomics, but these concepts apply also in lipidomics and metabolomics, but it's been determined that about 3/4 of the technical variability in proteomics experiments can be attributed to sample preparation. And so I think the overall objective is to kind of reduce the technical variability. You can start with sample preparation to reduce that. So starting in panel 1 on the left, if you look at traditional omics, often centrifugation and vacuum are used. If you view the arrows there, you can see that's the direction of the force. So in centrifugation, you have a transverse force, which in proteomics, this is showing the difference between using FASP. And this can be prone to a sporadic tearing of the filters, which can be detrimental in terms of a complete loss of the sample or clogging of the LC. With the vacuum, you can see that the force actually is applied to the bottom of the filter, which can result in the same negative results. In the middle panel in number 2, you can see on the left side we have a manual version of a pressure processor, and on the right side is the A200, which is the fully automated version. That is the M10 on the left. You see that you have control knobs on the front where you can control the pressure, and on the top you have a flow distributor manifold which can equally provide pressure to all 96 wells of a 96 well plate. So both of these instruments, they use the concept of positive pressure at which the force is applied at the very top of the liquid surface. And on panel 3, you can see that FASP, which is filter-assisted sample preparation, and PASP, which is the term pressure-assisted sample preparation. This term was coined by our collaborators at the University of Pittsburgh. You can see the reference at the bottom there for the ASMS poster. And in any case, for the 16 replicates you see there, the data for the number of peptides identified is nearly identical. There's also significant benefits in that with the positive pressure approach, you can robustly and expediently reduce the retentate volume. So this allowed them to reduce the overall number of steps in the process and therefore also reduce the time of the sample preparation. In addition to those benefits, there was been reported less filter failures due to the no transverse force. And then also the instrument on the right, if you look at panel 4, it has additional features. It has a touch screen on the front where you can programmatically write a method to dispense specific liquid volumes from up to 11 solvents. And there's also a pressure profile. So the pressure applied to the plate can be carefully controlled across a pressure gradient. And you can store and operate these multiple methods. And that system is also amenable to integration with a liquid handler for a fully automated workflow. In any case, you can see my LinkedIn profile there at the bottom. And we have a webinar next week, which would dive into these topics in significantly more detail if you have any interest. And with that, I'll leave it to the questions.
Thank you, John. I appreciate that. Thank you to all our speakers. So before we jump into the Q&A, I wanted to just thank our keynote speaker, Professor Oliver Fiehn, and especially our presenters, but also the team here that pulled this together within 1 week. So to the team, Aparna, Alice, Ivana, and Christina, and for Metabolomics Association of North America for endorsing us. I'm going to hand it over now to my co-host, Ivana Blaznovic, who's actually been collating with the team here all of your questions and will help them get answered. Ivana?
Thank you to all the participants for their questions. We have several of those, and we will try to get through all of them. So Uri Keshet has a question for Shenstock and regarding micro-LC. Uri, I will give you the chance to unmute yourself and you can ask the questions live. Is it working?
Ivana, you can ask the question and then perhaps Shen can reply.
Sure. So the first question was, how many MS2 scans can you get per one MS1 scan in data-dependent analysis on the micro-LC in regards to its narrow peaks? So that was the question Uri had.
Hi, in this experiment, the result was obtained by top 10 DDA with one fixed collision energy. So in this case, it's 10, but we can squeeze more because I think the cycle time is a bit higher.
Thank you, Shen. There was a follow-up question to this one. Is there any difference, did you notice any difference in compound stability due to less time at the high column temperatures?
The column, not really. The column temperature we use is like 35 Celsius degree. And it is right next, attached to the ion source, which is also very hot. So we only see like one case when we did repetitive injection, we see a slight, like less than 1 second retention time shift in the micro-LC, whereas in the UPLC is very stable. That could, like this could be one of the reasons, but it can also be the stability, like came from several factors, but which didn't affect our like data acquisition or downstream data processing as we saw.
One more question for you, Shen, regarding the number of features in the micro-LC. So Yasin noticed that you had decreased the peak width to 3 seconds, which can lead to detection of noise as peaks and splitting of peaks by the peak detection step. And the retention time tolerance was put to 0.3 seconds. Sorry. And the question is, because of the incorrect peak picking or the peak splitting, this could potentially result with like falsely increased in number of features. Is this something you have noticed, or how is the peak picking validated in micro-LC?
Cool. So first of all, because the peak has become narrower but much sharper, so actually we do see a like sharper and better peak shape that actually help with the peak picking. And we keep the same, and our peak picking and data processing parameters are based on what we observe in the raw data. As we can pull out the internal standard, we have a panel of 9 internal standard and verify to make sure we see all of that with a good peak shape, and our data processing parameters works fine for that. So the increased MZRT features, we do believe this is because of the higher intensity and better peak shape.
Thank you, Shen, very much for your answers. We will move to Oliver's talk. A lot of questions for Oliver as well. One in specific is regarding the MS-Dial version 4. And one of the users would like to know, one of the attendees would like to know if the newest version of MS-Dial is compatible with ion mobility data obtained in profile mode.
Yes.
So, Oliver, do you have information? Mm-hmm.
This is all from Hiroshi, and I saw that Hiroshi was online, but yes, it is.
Yes, Hiroshi is attending the conference. We are
So
very happy about that.
It's all really Hiroshi's work.
Yes. Okay, so Oliver, one more question for you. What does the community think about using commercially available software for lipidomic annotation, such as lipid search from Thermo? Anything to keep in mind when using such platform? We can also expand this question. Lipid search is from Thermo. I think there's a lipid annotator or lipid match from Agilent. So a lot of, I think companies, mass spec companies are coming up with their own software. So I think it's a good question to ask. How does the community think about this? How do you use it? And what are maybe some precautions that need to be made? Oliver.
I did answer this in my text. Did everyone see that?
Yes, we are just asking some of the most interesting questions live as well, in case not everybody is checking the
yeah, can you please read my answer, because I am already on another call.
Yes, yes, I will do that. Yes. Let's see the answer. I will have to get back to this one. Can't really find it right now. Okay. So there is a question.
I find it. Do you want to read it?
Yes, yes, please, Shan.
Regarding that, as answer from Oliver, I think it is great that the community has so many choices today with respect to different tools and softwares. I wish there were more side-by-side comparisons or workshops that would explore strengths and weakness of specific tools or algorithms. With respect to lipid analysis, there are quite a number of good tools out there because lipid MS/MS spectra can be predicted quite confidently, at least with respect to head groups and fatty acid chains. The question is, if there are more than one option for a lipid annotation, how is this reported? Are retention time models used to filter out in-source fragmentation, false positive MS/MS annotations? Does a software enable biology background information? And there are still ways to go. And check out MS-Style 4.0, accepted in Nature Biotechnology recently, with lots of more features for lipidomics analysis.
Thank you, Shan. We have two questions for our speaker, Dr. Marinka. And the first one is why apples were not compared with any other fruit like pear. And the follow-up question is the data that was shown today, has it been uploaded to metabolites? And maybe what is the accession number? Unmute, yeah.
This is it, unmute. So thank you very much for the questions. So I start with the first one. Yes, the dataset is uploaded into metabolites. I don't remember right now the number in my memory. You can download the paper. There are all data. They are uploaded also in the Dashin with several informations, and the files. Untargeted LCMS files, raw files, are uploaded into the metabolites. Then the other question was, I can also see here, there was someone that was asking if this study can be also extend to the wider pool of participants. Sorry, this suddenly pop up on my screen. So the 40 person with the mildly hypercholestemic volunteers, there was already a significant number to find differences. The main output for this study design was to check the cardiovascular risk factors, if they can be diminished through the additional 2 apples to the habitual diets. And this was the number of participants was designed during the step of the study design and the prediction of the outcomes. And this was succeed. So indeed, these 2 apples lower significantly one of the cardiovascular risk factors, which was cholesterol. And there is another paper saying about this. And it's already published, so I can provide maybe the link if necessary. And there was the question about the pears, why apples but not pears. So the apples are widely studied, and they have already proven health effects. Those were chosen for this experiment. Additionally, the apple are the most were widely distributed all over the countries. There was the participates here comes from UK, and apples are has pleasant taste, and they are easy. They can be eaten at any time. They can also be preserved for a longer time, definitely longer than pears. But of course, if there is any interest of the community, and especially the medical community, of checking the pears, such an experiment can be also added. In this case, we compared them with the control beverage, which was matched by means of energy and sugar in order to replicate the content of sugar and energy in this being at the same level, while the differences should be only to the presence of polyphenols and other apple constituents.
Thank you very much to our speaker. We have a question from our former colleague Sichuan Lai. She has a question for Oliver regarding the data normalization. So in specifically, her question is to Oliver, if you recommend normalization tool that is that can be applied to fecal samples specifically.
Oh, you guys are really tough with me because I'm still on the other call, of course. Let me see. Okay. This is a really tough question. I think we usually go for the total metabolome content for normalization. But one could also really think about distinguishing fecal water, that means excreted metabolites, from fecal solid matter, like bacteria and food. And I think any type of this kind of separation gets you closer to biological outputs. So we often go for excreted metabolites because these could be absorbed through the gut and interact with the human physiology, whereas the bacteria metabolome is a completely different question, right? So that's how I would think about it, but we usually go for the total metabolome.
And the software you would recommend for this, would you normalize it using?
Yeah, Excel, Microsoft Excel is a great software. Sorry.
Thank you, Oliver. In addition to this, there's also a surf normalization tool. You can just try it and see how it goes, also from Oliver's lab.
Their conference was just on the other end of this thing, and they ended up holding it, but a bunch of people.
Okay. We have a question for our speaker.
Oliver, I think you need to go back on mute if you don't mind. Yeah. Thank you.
Yeah. We have a question for our speaker, Dr. Machado. Since it's very difficult to obtain different tissue samples for metabolic profiling, especially for large cohorts, do you think that plasma metabolome information could be enough? And maybe one could avoid measuring all the other tissue?
Hi, can you hear me?
Yes.
Yeah, so there is a lot of research studying Roux-en-Y gastric bypass by metabolites from plasma. But our intention, we have the chance to collect gastrointestinal samples. So we decided to do metabolomics because we don't know exactly what are the gastrointestinal changes, the metabolomic changes. We already know that there is some gene alterations, especially at jejunum, but it's not fully understood how the surgery can impact the metabolism in gastrointestinal samples. So we also have other kinds of samples, but we decided to do gastrointestinal because we don't know any other paper that would do this, and is certainly something that is interesting to understand how are the mechanisms that the surgery impacts the type 2 diabetes improvement.
Thank you, Dr. Machado. We have
Ivana?
Yes.
What we were thinking was to swap out, just given that we're at the top of the end of our session here, seems like there's a lot of people still online. So, and there's still a lot of questions to be answered. So if people are interested, we're going to continue the Q&A session. So Arpana will be taking over. And I'd like to thank Ivana for taking this Q&A session, because I know you have other engagements right now.
Thank you, everybody.
Thanks, Ivana. Arpana?
Great.
Take over.
Okay.
Thank you.
Okay.
So I'll start off a question for John. If you're ready, automated sample prep is a must in a high throughput environment. However, some solvents may not be compatible with automation systems such as MTBE or chloroform. Are there any modifications such as fume hood use that are possible with your system that you presented today?
Yeah, so for the A200, which I presented, it does have the ability to handle those types of organic solvents. It uses a direct dispensing mode. It does have a small footprint, so it can fit in a standard fume hood. And then also there is a fan on the back that I didn't mention that could be vented to a the outside. So you can remove those. It is designed to remove those vapors from organic solvents, and it is specifically a lot of those solvents are very difficult to pipette with a traditional liquid handler. So I think that's where the benefit of the A200 is.
Great, thank you so much. I have another question for Crystal. If you're ready, can you tell us about ID challenges in plant metabolomics? This is actually a great question. Very interesting.
I know. I mean, I actually experienced that at the moment. The reason why I really wanted to reach out to you guys, because I know you're like at the forefront of plant metabolomics, and I've experienced like I have a lot of features, for example, and then they have the same molecular weight and similar retention times, but then it has no identification. And for now, I've been using the databases available, like from your lab, for example, and I've been downloading them and just analyzing my QC samples, and I'm intending to use my QC samples as my own in-house database. Like, do you have a comment about that? Or because that's like a problem that I have right now. I really wanted to ID a lot of compounds.
Yeah, I mean, we try to use as many libraries and also try to test free software that are like intended for in silico fragmentation and compound identification. So if that's ever of an interest, please feel free to reach out to our email at virtual.podium.2020@gmail.com. And yeah, we definitely have a lot of resources available between, just the hosts within the 5 of us. So thank you, Crystal, for your answer. I actually want to shoot back another question to John. Okay, so sample prep can be automated, including several washing steps. This can also be scaled to many samples, correct? Manual, only about 10 samples can be handled.
Yeah, so you have the ability to dispense 8 at a time, but it can do a 96-well plate at once. So you can handle 96-well plate in one run. So I just would comment with the multi-dispensing, you can dispense about 500 microliters of liquid in about 30 seconds to an entire 96-well plate. So it is pretty good with throughput for 96 at a time.
Great, thank you. Thanks, John. And I'm gonna bounce a question to Saiful Khan about L-Maven. Love the name. Very, very nice name. So L-Maven has a very nice user interface. Has the software been validated or compared against other metabolomics software such as MS-Dial, XCMS, MZMine, and which MS2 matching algorithms are applied in your software?
Thanks for the question. So we did a comparison against XCMS, but that was 2 years back, and L-Maven has significantly changed in these 2 years. So I think we need to redo that comparison, and I think we'll fare better this time. We also did a comparison with Skyline some time back, but I don't think that's a very fair comparison because Skyline is it tries to do a lot of things, and proteomics specifically. So but we're only focusing on metabolomics at the moment. That was your first question. What was the second question? I'm sorry.
What type of MS2 matching algorithms are applied
Right.
in LMAVEN?
Right. So we use something called a hypergeometric scoring, which is just it gives you a probabilistic score of how if two different spectra are randomly correlated, and then that's it gives you chance of that not being in that random correlation. So it's a little bit ad hoc, and I don't think it's documented anywhere, or there has been any publication around that. I'll find that out and let you know if you want to know. But I don't think there has been any documentation around that. It's a little ad hoc, and it was meant for a DDA workflow, which is already available on NVIDIA, and we've done some publication around that. But we're continuing to use that in DIA as well, because we've not explored a lot in terms of matching spectra for DIA fragmentation. Maybe we'll do some improvements on that front soon.
Great. So I just wanted to clarify, LMAVEN can also handle DIA data, or is it just swath data type?
No, so swath, we've not. So, okay, that's where the cross-platform thing, that's where the vendor-neutral thing comes in. So we only take in two different formats of files, that is MZML and MZXML. Now, if your DIA acquisition method is convertible to MZML in its current, in an equivalent format to swath, because we've only validated this on swath datasets. Mm-hmm. But if suppose vendor X publishes some new, releases some new DIA method, and that data can also be converted to MZML in an equivalent format, then we'll be able to deal with it without any problem. That Great. Depends on the conversion process, that's what.
Right. Okay, thank you. If we have time for one more question, I have a question for Annalise. So sphingolipids, like many other lipid classes, do not have standards available for purchase. Are you using in silico spectral libraries for sphingolipid identification, and how are your results validated?
We actually use deuterated standards available from Avanti. So
that's great.
Standards, yeah.
Awesome.
We can keep going up, Anna. We still have 80 people on the phone. We can go till 1:30 at least. If we still have questions to be asked. If not, I know that there's Hardik Shah is in the audience who's raised his hand, so we could potentially take live questions. But if you have some questions there, we can take those first.
Yeah, there's one question for Nicholas. Mm-hmm. SRMs are being validated based on the features and their %CV based on Progenesis peak picking software. Does other software show similar results? And how important are IDs in the context of SRMs?
Okay, so far we have been using Progenesis QI for doing the peak picking process, and now we want to compare these results with CMS or other software, but I cannot give you a certain answer because Progenesis QI is the only option I have been used so far.